Imagine an issue that, well, costs economies, millions, can shift entire ecosystems, and even carries a direct threat to public health. Sounds like something we all need to know about, right? Welcome to the deep dive. Today, we're plunging into a topic you might not have considered much: pregnancy loss in animals. It's far more than just a single biological event. It's actually a critical factor with these massive implications that really touch us all. Our mission for this deep dive is to sort of distill the most important insights, uncover some surprising connections, and highlight why these topics, which might seem niche, are profoundly relevant to you and, the broader world. We're going on an in-depth exploration into the causes, the diagnosis, and the management of pregnancy loss in two distinct yet incredibly important groups: camels from the Camelidae family and deer from the Cervidae family. The core problem is similar, yeah, but the context, the impact, and, well, the solutions often diverge quite dramatically. So when we talk about pregnancy loss, we're really looking at a spectrum, aren't we?
Oh,
It can range from, say, early embryonic death, where the embryo is lost even before its vital organs start forming organogenesis, I think it's called,
that's right, organogenesis, often just reabsorbed, gone without a trace sometimes.
all the way to late-term abortion and stillbirth. So given this wide range, what makes figuring out the specific cause such a notoriously challenging puzzle for experts?
It's a truly multifactorial problem. I mean, you're dealing with this complex interplay of infectious agents, bacteria, viruses, and you name it, genetic factors, nutritional issues, toxins in the environment, even just broad environmental stressors. What's fascinating, and it also complicates things, is that often an underlying issue, let's say poor nutrition, can make an animal much more vulnerable to an infection that then causes the loss.
Oh, okay.
So it's rarely just one single thing. It's usually an intricate web.
Right. So we're talking about animals as different as camels, out in the desert, and deer maybe in the woods behind your house, but they face surprisingly similar challenges in carrying a pregnancy to term. It's quite a convergence.
It really is.
Let's try and unpack some of these hidden triggers then, starting with the microscopic world, the infectious agents.
That's a good place to start. The broad categories of causes, infectious and non-infectious, they're shared, yeah. But the specific bugs, the viruses, the bacteria, and how they impact the animal, that often highlights the unique biology and the specific environments these animals live in. So among the infectious causes, we encounter a whole host of pathogens.
And some of these microscopic saboteurs are particularly sneaky, aren't they? Like brucellosis in camels,
That's bacterial, right?
Urticella
Brucella abortus and B. melitensis.
Exactly. Those are the main culprits in camels. And the numbers can be significant. Some studies show up to, 32.5% of aborted camel fetuses testing positive?
Wow.
But here's the kicker, the real challenge. Brucellosis is often subclinical in the pregnant dam.
Meaning she seems fine.
Completely fine. The abortion itself, that event, might be the only clinical sign you ever see.
That must make surveillance incredibly difficult. It creates a major surveillance headache. If you're just waiting for animals to look sick, you're missing the boat entirely with brucellosis. The key insight here really is that every single abortion has to be treated as a potential warning sign for herd-wide infection. Otherwise, it just spreads silently.
Okay. Then there's trypanosomiasis, or sura, also in camels. Now, this isn't a bacterium, right? It's a protozoan parasite.
Correct. Trypanosoma evansi. It's transmitted by biting flies like horseflies. And this one typically causes late-term abortions, usually between the 8th and 11th month of pregnancy in camels. It can cause some pretty significant damage, pathology in the fetus itself.
And you mentioned a unique viral twist, something about a horse virus.
Ah, yes. Equine rhinitis virus, ERV. Normally, in horses, it's a respiratory thing like a cold, mild symptoms. But in dromedary camels, it specifically targets the genital tract. And it can cause these large outbreaks, what we call abortion storms.
Seriously, a horse cold virus causes abortion storms in camels.
Yep. It's a really fascinating example of, interspecies transmission and how a virus can find a totally new way to cause severe disease in a different host.
That is wild. Okay, moving over to deer then. They face their own specific viral threats, you mentioned, mainly from arboviruses, viruses spread by insects?
Indeed.
Mm.
Two big ones are episodic hemorrhagic disease, EHD, and blue tongue virus, BTV, both transmitted by these tiny biting midges, chylicoids.
Right.
Now, these often cause outbreaks of acute death in deer populations. You might hear about those die-offs. But they're also responsible for abortions and some really severe congenital malformations in the fawns that do survive pregnancy. Things like hydranencephaly.
Hydranencephaly, what's that?
It's where the cerebral hemispheres of the brain basically don't develop properly and are replaced by fluid-filled sacs. It leads to things like ataxia, severe wobbliness, and blindness, obviously not compatible with survival in the wild.
Grim. And then there's chronic wasting disease, CWD. That's the prion disease, right? Like mad cow disease, but in deer.
Exactly. Caused by these abnormally folded proteins. Now, CWD is a bit different. It's not a direct abortifacient protect. It doesn't usually cause the doe to immediately expel the fetus.
Okay.
Instead, its impact is more insidious, more subtle. It affects fetal development. We see, on average, about a 1% lower birth weight in fetuses from CWD-positive does.
1% doesn't sound like a lot on its own.
It doesn't, but lower birth weight is directly linked to reduced fawn survival, especially through that critical first winter. And maybe more importantly, if we connect this to the bigger picture.
Yeah.
CWD drastically reduces survival for both fawns and the adult does themselves. Infected deer are up to 4.5 times more likely to die annually compared to uninfected deer in the same population.
4.5 times.
Yeah, so it profoundly affects the population's ability to sustain itself. It acts like this powerful, slow-burning fuse, limiting the population over time.
Okay, so shifting away from infections now, let's talk about the non-infectious causes, the environmental stuff, the physiological stressors, twinning in camels, for instance, that came up in the research.
Yes, that's a major non-infectious cause. One study pegged it at 13.1% of diagnosed abortions.
Yeah.
Usually happens between months 5 and 9 of gestation.
And why is that?
It's basically uterine capacity. The camel uterus, well, it's generally not well suited to supporting two fetuses all the way to term.
Yeah.
There just isn't enough space or placental support.
Makes sense. And nutrition, that seems like a common thread.
Absolutely. Nutritional deficiencies are huge and often foundational. Lack of essential vitamins like A or E or trace minerals, selenium, iodine, or critical ones can cause problems in both groups. For deer, you could argue that nutritional stress is probably the single most important factor overall. Poor body condition in the doe doesn't just directly lead to fetal loss, it also makes her significantly more susceptible to infectious diseases. See how they're intertwined?
Yeah, definitely. It weakens their whole system.
Precisely.
And it's not just about lacking nutrients, right? There are also environmental toxins, contaminants, things getting into their environment.
Yes, that's an increasingly concerning area. Beyond naturally toxic plants, things like locoweed or hemlock, which animals might eat, we're now really looking hard at anthropogenic contaminants, pollutants from human activities.
Okay, like what?
Well, there was this one landmark study, really eye-opening. It looked at the neonicotinoid insecticide imidacloprid in wild white-tailed deer.
That's a common pesticide, right?
Very common.
Yeah.
And they found that the levels present in these wild deer, just from an environmental exposure, were, on average, 3.5 times higher than the levels known to cause adverse effects in lab studies with deer.
3.5 times higher than the problem level. Wow.
Yeah, and these weren't trace amounts. These were field-relevant levels directly impacting thyroid hormones, body weight, and ultimately fawn survival and mortality.
That really drives home the connection, doesn't it, between what we put into the environment and wildlife health?
It absolutely does. These aren't just abstract chemicals out there. They're directly compromising the health and the reproductive success of deer in their natural habitats. It's a clear link.
And finally, just plain old stress, that can trigger abortion too?
Definitely. Things like transport, especially long or rough journeys, extreme weather events, improper handling, even something called overbreeding in camels, basically, mating too frequently, leading to uterine inflammation that can all trigger abortions. The body just says, nope, can't handle this pregnancy right now.
So it's this incredibly complex mix of potential causes, tiny viruses, environmental toxins, nutrition, stress. How on earth do experts figure out what actually went wrong in any specific case? It sounds like some serious detective work.
It absolutely is detective work, yeah. And you hit on a key point. How do you get a definitive diagnosis when the evidence, the fetus or placenta, might be really degraded?
Right. Decomposition must be a huge issue.
It is. Autolysis, the natural breakdown of tissues after death, can mask the crucial lesions, the telltale signs, and it can destroy the pathogens themselves. Makes the job significantly harder. But a systematic approach really does increase the chances of getting an answer.
Okay, so walk us through that systematic approach. From the field, when an abortion is discovered, to the lab, where do you start?
It really has to start with a thorough field investigation. You need a detailed history from whoever owns or manages the animals, vaccination records, any recent animal movements, what they've been eating, any potential stressors. Then you examine the dam, the mother, look for any signs of illness herself, like fever, discharge, or, importantly, a retained placenta.
And then collecting samples, that sounds critical and probably tricky.
It is critical and needs to be done right. Ideally, you submit the entire fresh fetus and the entire placenta. Chilled, never frozen. Freezing damages tissues for microscopic work, and get it to the diagnostic lab fast.
Why the emphasis on the placenta?
Because the placenta is often ground zero. Many pathogens cause very characteristic inflammation or damage there, giving us vital clues even if the fetus looks relatively normal.
Okay. And if you can't send the whole thing?
Then specific tissues are the next best thing. Sections of the placenta are still top priority. Then lung, liver, spleen, kidney, brain, and importantly, stomach contents, the abomasal contents. You need some samples fresh, kept chilled, for microbiology and molecular tests like PCR. And you need other pieces fixed in formalin for histopathology, the microscopic examination.
And you mentioned maternal blood samples, too.
Yes, crucial. Paired serum samples from the dam. One taken right around the time of the abortion, the acute sample, and another one taken 2 to 3 weeks later, the convalescent sample. Comparing antibody levels between those two samples is key for serology. A significant rise in antibodies tells you the mother was actively fighting off a specific infection around the time of the loss.
Got it. So once those precious samples arrive at the lab, that's where the high-tech detective work really kicks in.
That's right. Multiple sections of the lab get involved.
Where's the first stop, usually?
Often, it's the microbiology lab. They'll take the fresh tissues, lung, liver, placenta, stomach contents, and try to culture any bacteria or fungi that might be responsible. They use specialized growth media to try and isolate common culprits like Brucella, Listeria, Salmonella, Campylobacter.
Okay. And what about the things that are hard to grow, or maybe you need a faster answer? That's where PCR comes in, right? You mentioned that for viruses.
Exactly. PCR polymerase chain reaction has been, well, revolutionary for abortion diagnostics. It's incredibly sensitive and specific. It detects the pathogen's DNA or RNA directly from the tissues.
Even if the bug itself is dead.
Even if the organism is no longer viable or if it's only present in very low numbers, it's become a primary tool for diagnosing many viruses like EHD or BTV, and also for those difficult-to-culture organisms like Trypanosoma or Coxiella burnetii, the Q fever agent. Many labs now run these comprehensive PCR panels that can screen for a whole suite of potential abortifacients all at once from a single sample. Gives you rapid, detailed answers potentially.
Okay, so culture, PCR. Then there's serology, looking at the antibodies in the blood samples you mentioned.
Right, the paired serum samples from the dam, or sometimes fluids collected from the fetus itself. If we see that significant rise in antibody levels, the titer between the first and second sample, that's strong evidence the dam mounted an immune response to a specific pathogen right around the time the abortion occurred.
So it points the finger at that pathogen.
It strongly suggests it was involved, yes.
And finally, histopathology. Looking at the tissues under the microscope.
Absolutely crucial. It starts with a detailed gross necropsy examining the fetus and placenta with the naked eye. Are there obvious lesions? A thickened, leathery placenta might suggest brucellosis. Little white spots, necrotic foci on the liver could point to a herpes virus infection. Then, histopathology takes thin slices of those formalin-fixed tissues, stains them, and examines them under the microscope. This lets the pathologist see characteristic tissue damage, inflammation patterns, cell death, and sometimes they can even visualize the organisms directly within the lesions using special stains. It's all about putting together the complete picture using every piece of evidence available.
Okay, so we've covered the what the cause is and the how, the diagnosis. Now, what does this all mean for actually managing these animals? This seems to be where the story of camels and deer really takes dramatically different turns. They have such different roles in our world.
That's exactly right. If you connect this back to the bigger picture, the fundamental goals for managing these two groups are almost opposite. And that difference shapes pretty much every single management decision. For camels, generally speaking, we're trying to prevent every single loss, whereas for wild deer populations, sometimes reproductive failure is actually part of a larger ecological management strategy.
Okay, let's start with the camels then. The goal is protecting each pregnancy, maximizing economic output, food security. Every lost pregnancy is a direct economic hit, right?
Absolutely. For pastoralists and farmers relying on camels for milk, meat, transport, or income, each pregnancy is incredibly valuable. So the strategies are intensive, focused on individual animal health and herd productivity.
So what does that look like on the ground?
Well, first and foremost is biosecurity. Strict protocols. Quarantining any new animals brought into the herd, controlling movement between groups, really rigorous hygiene, especially around births, And critically, proper disposal of any aborted fetuses or placentas, burying deeply or incinerating them to prevent the spread of dangerous pathogens like Brucella.
Makes sense. And I assume vaccination is a big one.
Yes, where effective vaccines exist, targeted vaccination programs are vital, especially against prevalent diseases like Brucella, using established vaccines like Strain 19 or Reverin 1, depending on the region and regulations. This is about preventing outbreaks before they start.
And monitoring the herd. Surveillance.
Constant vigilance, regular serological testing, blood tests to check for diseases like brucellosis. In areas where it's common, you might see test and slaughter policies, removing infected animals, or sometimes test and segregate, keeping infected animals separate to protect the rest of the herd.
And if an infection does get in, what about treatment for camels?
It depends on the cause, of course. If it's bacterial and caught early enough, targeted antimicrobials based on culture results might be an option, along with supportive care for the dam. But just as important is addressing any underlying issues contributing to susceptibility, like those nutritional deficiencies we talked about.
Right. Which brings us to husbandry.
Exactly. Optimized husbandry is paramount, ensuring balanced nutrition, especially in that critical late stage of pregnancy. Avoiding stressful events like unnecessary transport. Practicing proper mating management to prevent that uterine inflammation from overbreeding. And just ensuring clean feed and water sources. It's a whole system approach aimed at safeguarding each pregnancy.
Okay, now let's pivot completely. Wild deer populations, where the goal is often the opposite, managing reproductive outcomes, sometimes even limiting them. Why?
It's all about ecological balance. For many wild deer populations, especially white-tailed deer in parts of North America, the management goal is to regulate their numbers to align with what the environment can actually support the ecosystem's carrying capacity.
To prevent overpopulation.
Precisely. Unchecked deer populations can cause significant damage. They can devastate forest understories, preventing tree regeneration, which impacts other wildlife. They can increase tick populations and Lyme disease risk. They cause more vehicle collisions, so the goal becomes preventing overabundance.
So how do wildlife managers achieve that in a wild, free-ranging population? It sounds much harder than managing a herd.
It is different, for sure. The primary tool for population control in most situations is lethal removal, typically through regulated public hunting. The aim is to maintain deer densities at or below the ecological carrying capacity. This not only protects the habitat, but also reduces the transmission rates of density-dependent diseases.
Okay, hunting. What else?
Habitat improvement is also a key strategy, but it works in a slightly counterintuitive way regarding reducing overall numbers long-term. Enhancing forage quality and availability through things like prescribed burning, specific timber harvesting practices, or planting food plots directly improves the body condition of the does.
Which you said leads to better reproductive success.
Initially, yes. Healthier does have more fawns that survive. But the goal of habitat improvement in a management context is often to support a healthier, more resilient deer population at the desired density level set by hunting or other controls. Better habitat means the ecosystem can better support the target number of deer without degradation. It creates that pathway. Better habitat, better nutrition, healthier deer, lower non-hunting mortality, including less loss from disease or starvation.
What about fertility control? That gets talked about a lot as a non-lethal option for deer. How effective is that?
Fertility control. It's a complex tool, and honestly, it has significant limitations for managing large, free-ranging wild populations. Things like immunocontraceptive vaccines, PZP, or Gonacon, or surgical sterilization, they can work, but well, first, they usually require a relatively closed population where deer aren't constantly moving in and out. Second, you need to treat a very high percentage of the females, typically 70 to 90% year after year, which is logistically incredibly difficult and expensive.
Oh,
We're talking estimates of maybe $600 to $800 per doe treated.
wow.
And critically, it's slow-acting. It only prevents future births. It does nothing to reduce an existing overpopulation problem quickly. Agents that might induce abortion, contragestation agents, they're generally problematic due to the risk of just rebreeding later in the season, or animal welfare concerns if administered too late in pregnancy. So while it might have niche applications, fertility control is rarely the silver bullet for broad-scale deer population management. So this raises, I think, an important question. We've got these dramatically different management goals: save every camel pregnancy, manage deer reproduction. But what unifies these challenges? What's the critical common thread that really impacts all of us, regardless of whether we're talking about livestock or wildlife?
It has to be the public health risk, doesn't it? You mentioned pathogens. Many of these things that cause abortions in animals can also infect people. Zoonotic diseases.
Exactly that. That's the crucial overlap. Many of these abortifacient pathogens are indeed zoonotic. They pose significant risks to human health.
Like what specifically? We mentioned brucellosis.
Brucella spp is a major one, causing undulant fever in humans, a potentially chronic, debilitating flu-like illness. Then there's Coxiella burnetii, the cause of Q fever. This one's particularly insidious because the placenta, even from an animal that seems perfectly healthy at birth, can be loaded with bacteria and highly infectious through aerosols.
Just breathing it in?
Potentially, yes. Also Leptospira, SPP, which can cause kidney failure, liver damage, meningitis in humans, and Toxoplasma gondii, a toxoplasmosis parasite, which is especially dangerous for pregnant women, potentially causing severe birth defects, and also for anyone with a weakened immune system.
So who needs to be most aware of these dangers? Who are the high-risk groups?
Well, certainly veterinarians, farmers, camel pastoralists, slaughterhouse workers, anyone handling livestock directly, but also, importantly, hunters, field dressing deer carcasses, and wildlife biologists handling animals or samples.
How does transmission happen?
It can be through direct contact with infected tissues, especially aborted fetuses, placentas, or uterine fluids, inhalation of aerosolized bacteria, like with Q fever, or even consuming unpasteurized milk or undercooked meat from infected animals.
Which means prevention for people handling these animals or materials is absolutely critical.
It is paramount. We really have to underscore the vital importance of using personal protective equipment, PPE. Gloves are essential, masks, eye protection, especially when assisting with births or handling aborted materials from any ruminant species, whether it's a camel, a deer, a cow, a sheep, or a goat.
So really, an abortion event isn't just an animal health issue or an economic issue or an ecological one.
It's a public health necessity to handle it correctly. It demands that integrated One Health approach, recognizing the interconnectedness of animal health, human health, and environmental health. We can't manage them in isolation.
This deep dive really highlights that fundamental paradox, doesn't it? An abortion is this economic disaster you need to prevent at all costs in camels, impacting food security for potentially hundreds of thousands of people. But then reproductive failure, or at least controlled reproduction, can actually be a desired ecological management outcome in overabundant deer populations, helping prevent habitat damage and protect biodiversity.
It's a stark contrast in objectives. Yet, despite those differences, the shared challenge remains crystal clear.
Oh,
Both scenarios are deeply intertwined with our own health because of that pervasive risk of zoonotic diseases.
yeah.
The health of our animal populations, whether they're valuable livestock or free-ranging wildlife, is just inextricably linked to our own well-being and the health of the environment we all share.
So what does this all mean for you, our listener? Maybe the next time you hear about an animal disease outbreak or see deer in a park, or even think about where your food comes from, consider that unseen web of connections. From a remote camel herd facing economic hardship in the desert, to those deer potentially reshaping the forest in your local area, and the surprising, sometimes invisible, risk of diseases jumping between animals and people. The health of our animals, livestock, and wildlife that really is tied up with our own health and the planet's health. There's always more to learn, and those connections, well, they're often much closer and more complex than we might think, demanding our attention and definitely a holistic view.